Greetings and welcome to episode number 10 of Repligen's 2022 webinar series. My name is Wesley Straub, Marketing Director here at Repligen. Of the 10 webinars that we've done this year, this is our fifth one on intensification because it's such an important topic. If you happen to miss the previous four, in those four, we covered first a case study on intensification of vaccines, and then a case study on intensification of viral vectors. On the third one, it's kind of a brief history of time. We did a 15-minute encapsulation of about 15 years of history and knowledge on intensification. And then in the fourth one, we covered a look at the workflow from a working cell vial all the way through the seed train N-1 bioreactor out to the end production bioreactor, and where in that workflow you could intensify and why you would intensify it. Today's webinar moves us a little bit more into the future and on the technology side of things to how you can intensify faster and easier, right? And to tell that story today, we have two great speakers. First, Charles Hill is one of our exceptional field application scientists here at Repligen. Charles is extremely knowledgeable, very friendly, and always willing to share that knowledge. And then we're also very lucky to have a customer guest speaker, Emily Rittershouse. Emily did these experiments with her team in her lab, and so she's going to talk about the motivation for these experiments and then walk us through the data. Before we get started, there are three housekeeping items that we quickly need to go through. First, this webinar is being recorded so that it can be viewed by those who could not join us today on demand. Second, this is an interactive platform. You can ask a question at any time if you open the link down below. We accumulate the questions over the course of the webinar. You'll see actually an upvote function. You can use that upvote function at any time as well. It's really helpful because we typically get more questions than we can answer in the allotted time frame, and it helps us prioritize those. So definitely pose your question, and they'll all be answered in a live discussion with Emily and Charles after the presentation. Poll question. It'll take you about 15 seconds to answer it, but it really helps us guide the content of future webinars towards what's most interesting to you. And with that, it takes care of all my housekeeping items and background. So with that, I'm going to hand it off to Charles and Emily for today's webinar titled Evolution of Upstream Intensification, Optimizing Perfusion Processes with PAT.
Thank you, Wes. Hello and welcome. My name is Charles Hill, and I am an upstream field application scientist here at Repligen. I would like to thank you for joining us for another webinar in our Repligen 2022 series. Before I hand it off to our esteemed speaker today, I would like to set the stage for our topic of discussion, which is the evolution of upstream intensification through optimizing perfusion processes with process analytical technologies. Here at Repligen, we are a leader in upstream process intensification with over 15 years of experience providing innovative technologies for upstream intensification that are scalable from benchtop all the way up to large-scale manufacturing, while staying dedicated to providing the highest quality products. Pictured on the right are two of our leading cell retention technologies utilized for upstream processing. Our first is a crossflow TFDF technology that is used in the viral vector space, exosomes, and other vaccine productions. Our second is the XL-ATF. This technology is utilized in a wide variety of modalities, including proteins, monoclonal antibodies, cell therapies, and so on. Our upstream cell retention devices have a broad platform adoption, with over 500 sites globally utilizing our technologies. These include over 40 commercial processes with hundreds of programs in early and late stage development, with over 25 cell lines that have been proven to be intensified. Some recent applications with XL-ATF are highlighted on this slide, which includes Genentech utilizing intensification to meet demands at smaller scales, providing a more nimble manufacturing, as well as WuXi achieving 50% capacity increase from intensifying 5 to 6% of their bioreactors. And there are many more that aren't listed here. With the push into process intensification across a wide variety of modalities comes the question of how can these processes be more robust and more efficient through incorporating things such as PAT tools. Where does PAT fit in process intensification? There are many process parameters and quality attributes that must be monitored and controlled during bioprocessing. These include things like metabolites, viable cell densities, viability, titer, and so on, with many technologies that are on the market that can solve these challenges. By leveraging PAT with process intensification tools like the XL-ATF or CrossFlow TFDF, you can drive robust, efficient, and scalable platforms for different therapeutic workflows. And today our guest will talk about focusing on utilizing capacitance probes and upstream intensification to highlight the benefits of the evolution in this intensification. And with that being said, I would like to introduce our speaker, Emily Ritterhouse. Emily is a senior scientist in the Upstream Process Development Group at Bristol Myers Squibb in Devens, Mass. Emily has worked at Bristol Myers Squibb for the past 4 years, performing different functions such as leading upstream process development for different assets, characterization, and technical transfer for both fed batch and perfusion processes. Emily has a bachelor's degree in microbiology from McGill University and a PhD in biomedical sciences from the University of Massachusetts Medical School. And today, Emily is here to share how BMS has applied process analytical technology to upstream intensification towards developing a monoclonal antibody N-1 platform process. Please take it away, Emily.
Thank you, Charles, and thank you, Repligen, for having me today. My name is Emily Ritterhouse, and I've been a scientist at BMS for about 4 years now. And I'm going to be presenting our work today on N-1 perfusion platform development using PAT for biomanufacturing. And so I'm just going to start off with a quick outline. I'm going to briefly introduce a few different subjects that's important for this work. Firstly, fed batch process intensification and what that is. Then perfusion N-1, give some background on our capacitance probes, and give some background on why we want to have some platform development work for our N-1 perfusion processes. Then I'll be getting into our results with this work. So firstly, our offline to online VCD correlation, followed by a media usage optimization study, followed by our CSPR optimization study, and then our inoculation density optimization, and finally followed with our scale-up capacitance performance, and finish with our summary and conclusions. So intensified production is essentially an increase in inoculation density in fed batch end stage processes, which can increase volumetric productivity. So for example, a conventional fed batch might be something where you inoculate at 1 million cells per mil versus an intensified fed batch, which could be inoculated anywhere from 2 to 8 million cells per mil, or even up to very high cell densities of 10 to 20 million cells per mil. And doing so can double fed batch titers. So it's really something that we have wanted to work on at BMS, and we have worked on at BMS, and we've been very successful in increasing our titers by increasing our inoculation density. However, having a high inoculation density requires a high final VCD N-1 process. And that is where our perfusion N-1 comes in. So perfusion N-1 can achieve greater than 100 million cells per mil, allowing us to inoculate at these very high cell densities of greater than 10 million cells per mil. And our perfusion N-1 into an intensified fed batch is cost effective in terms of the dollars per grams of antibody produced compared to a traditional fed batch process. And so just to give a little bit of background on our perfusion rate and what we mean here by that. So Perfusion N-1 processes utilize a filter. For all of our studies here, we use the Repligen ATF2, which essentially we just use to remove spent media and continually add in fresh media at a defined rate. And this defined rate is our perfusion rate. The perfusion rate efficiency is very important for a number of things. Probably most importantly is it's important for achieving a high final VCD and high viability. So high viability is very important for the N-1 step. It's also very important for cost effectiveness as well as efficiency for media preparation and storage. And common methods for defining a perfusion rate are typically a fixed volume. For example, vessel volumes per day, also known as VVD. Alternatively, you could also use a cell-specific perfusion rate, abbreviated as CSPR, which can also be advantageous. And it's advantageous just because if you look at a volume-specific perfusion rate, while it has a straightforward implementation, you can pretty easily define where you want to start off just based on doubling times and where you want your final VCD to be. You can easily have a straightforward implementation of a volume-specific perfusion rate. However, you may be over-perfusing your media, which is inefficient, or at certain stages you may be under-perfusing media, which could negatively impact your growth and viability. And optimization of this may be time intensive. So something that we have been working on at BMS and in this work here is to implement a cell-specific perfusion rate. And this cell-specific perfusion rate supplies media on a per cell basis. And theoretically, this precise amount of media required will be able to support cell growth at all VCDs. And it should be cost effective for media usage. And something that can make it even more efficient is using an inline monitoring of VCD, for example, a capacitance probe. Integrating this into the system greatly simplifies your CSPR optimization. And so just to give a quick background on capacitance, so capacitance probes allow for real-time monitoring of VCD in your bioreactor. So essentially how it works is cells, intact cells with the intact plasma membrane will act as a small capacitor in the presence of an electric field. And these measurements taken by the probe are proportional to VCD with accuracy up to very high cell densities. So it's great for a perfusion N-1 process. These probes can be integrated with bioreactor control systems, for example, Delta V, to automatically adjust the CSPR based on VCD. And it's also a GMP compatible technology that's already in use in pharmaceutical manufacturing operations. And so finally, I just want to introduce the idea of platform development and why it's useful. So our goal here is to develop a platform N-1 perfusion process by testing similar conditions in multiple clones. And so a manufacturing platform is a framework for developing production processes for similar modalities. And in this case, we're using monoclonal antibodies. So we want to be able to demonstrate applicability in many clones to build confidence in future applications. So anytime we bring in a new antibody, we can be confident that this platform will work well with this new program. So an effective cell culture platform should define a starting set of cell culture conditions that work well for most clones. And we wanted to develop, a workflow for efficiency, for improved performance, and/or robustness. And so in this study, we applied an inline capacitance controlled CSPR to 6 antibodies in development and used this to establish a platform workflow. So essentially what our workflow looks like is we want to be able to take a new antibody into our development group, do 2 to 3 experiments with it, and then have it just be ready for scale up and implementation into our pilot scale or manufacturing scale. And now getting into some of our results. So our first goal was to determine the online to offline correlation for each clone up to high cell densities. And what I'm showing you in these panels here, A through F, are 6 different antibodies that we used in this study. And we're showing their online capacitance measurement permittivity correlated with the offline VCD, which we measured using the ViCell. And basically what we see is that, regardless of the initial seeding density from low to medium seeding densities, and then regardless of the final VCD, so we saw anywhere from 80 to 130 million cells final VCD, cells per mil final VCD, we were able to see that we got a very good correlation between the offline and online permittivity, which was great. We also noticed that the slopes varied for each antibody. So this is a linear relationship in the N-1. And so what that means is that the slope is basically specific to each clone. And so this is something that needs to be done when you bring in a new antibody to your program, is to determine this online to offline correlation. And we're calling this the capacitance correction factor, or CCF. And essentially what it means is that the permittivity multiplied by this capacitance correction factor is going to give you your VCD. And next, what we wanted to do was show that we could quickly optimize our perfusion rate. And what we found was that the capacitance controlled CSPR reduces media usage in our perfusion N-1 bioreactors. So what I'm showing you here in panels A and B is antibody 1, and then in C and D is antibody 5. And in each case, we're comparing a online controlled CSPR versus a fixed volumetric perfusion rate. And the volumetric perfusion rate, we utilized the doubling time and the final desired VCD to estimate what that perfusion rate should be. And for the capacitance method, we just utilized the online VCD and the CSPR of either 0.03 or 0.04 nanoliters per cell per day, depending for, respectively for antibody 1 or antibody 5. And we saw that both produced very similar final VCDs and viabilities. For antibody 1, it was both around 50 million cells per mil final VCD. For antibody 5, it was around 100 million cells per mil final VCD. And for both of these cases, the capacitance controlled CSPR resulted in about 25% decrease in media usage with no optimization. So essentially, we were able to just plug and play with the probes once we had the capacitance correction factor. And we were able to see a large decrease in the media usage. Next, our goal was to determine the optimal CSPR quickly. And so an optimal CSPR perfuses just enough media to support high VCD and high viabilities. So for what I'm showing you here, the data slides, so we have in panels A and B, we're showing you antibody 3. So we're looking at the cumulative media perfused, as well as the VCD and viability. And then in C and D, we're looking at antibody 5 for the same parameters. So we could see that 0.04 nanoliters per cell per day CSPR had similar VCD at 0.06 while using 30% less media. And this was true for both antibody 3 and antibody 5. Also, we saw that when we tested CSPRs below 0.04 nanoliter per cell per day, we negatively impacted the VCD and viability. So just looking at all of the antibodies tested in this study, 4 of the 6 performed well with the CSPR of 0.04 nanoliters per cell per day, regardless of doubling time, inoculation density, and total process duration. So we're able to establish a platform CSPR of 0.04 nanoliters per cell per day. Next, we wanted to be able to determine our optimal inoculation density quickly. And so basically what we're looking at here, we're looking at one antibody in panels A, B, C, and D, but we are now looking at 5 different inoculation densities that we tested. And in panel A, you can see a dotted line, which is what our target final VCD was. And so final VCD can be difficult to predict at these high cell densities due to a slowing of doubling time, as you can see in panel D. But we wanted to design one experiment where we could test a span of inoculation densities like all at the same time. So we had 5 reactors in parallel. We're able to show that actually our highest inoculation density of 3.9 times 10 to the 6 million cells per mil was able to achieve a target final VCD, our target final VCD, in a shorter duration with lower media usage. And so this platform development workflow, we're then able to show that we can identify an optimal CSPR and an inoculation density, all within a short set of experiments. And then our next step is really to just show that we are able to scale up this process well into our manufacturing scales. And so our goal here is just to implement our capacitance perfusion control at pilot and manufacturing scale. And so here we're showing scale-up performance of MABS 4 and 5 at 5 liters, 200 liters, and 500-liter scales. And just wanted to note that we're using the same capacitance probes across all scales. And we're using the Repligen ATF2 at the 5-liter scale, and then the ATF6 at the 200 and the 500-liter scales. And basically what we see is that we're able to achieve very similar VCDs and viabilities using an inline capacitance controlled CSPR and the same capacitance correction factor and process determined at the 5-liter scale. So we were able to successfully implement these into our GMP facility. So just to summarize these slides I presented today, basically we're able to demonstrate integration of an inline capacitance probe for multiple monoclonal antibodies. We're able to show that our permittivities had a strong correlation with VCD up to very high cell densities. An application of the inline capacitance probe made our N-1 processes more efficient and faster to develop. We're also able to show that media use was reduced by approximately 25% when compared to a platform volume-based perfusion strategy. And our platform development workflow was streamlined by using the CSPR as a single variable that adjusted perfusion rate when the growth rate shifted. And this technology enabled multiple successful large-scale N-1 perfusion runs, including several in a GMP manufacturing setting. And so just to finish off, I'd just like to acknowledge a lot of people in our upstream biologics group had a hand in all of these studies. So I'd just like to acknowledge the whole group, as well as the co-authors on this paper. And please look it up if you'd like to read more. There's a little bit more data in there if you're interested. But specifically, I'd just like to thank Matt, Jim, Shin, Charles, Jeff, Mike, ZJ, and Anurag for all of their work on this subject. And thanks you to Repligen for inviting me.
Okay, thank you, Emily. And on the topic of intensification, and because we have such great speakers and Charles and Emily, as expected, we have several questions. So right now in the queue, we have 7 questions. Everyone in attendance, you're welcome to keep submitting questions over the next couple of minutes. But Sydney has 2 upvotes to her question. So we're going to start with Sydney's, right? And so Sydney's question is, can you expand on how long it took BMS, Emily and team, to develop a platform, an N-1 platform fusion process, as well as how quickly it can be applied to new molecules? So it sounds like it's like maybe how long did it take to get your first N-1 process up and running, and then maybe how long it took after you learned a couple things to get, the subsequent ones up and running.
Yeah, that's a good question. So, I mean, we actually started doing this perfusion N-1 development before I joined BMS even. So I think from the original time to the time we got a platform process was probably a few years. However, a lot of the initial studies were really just, initial studies. And I think by the time we were sort of rolling and had a good thing going, it took us about, I would say, probably about 1 year to develop a platform process.
Okay. And then I think, so after that, so if you have your platform process, right, how long is it roughly, like if a new molecule comes in? Well, that's like when you say new molecule, let's say it was a new antibody, right? How long to move on to a new antibody rather than like, a whole new protein or something completely different?
Yeah, if we had a new antibody come in, I think, best case scenario, if we had a very short timeline, not best case, but if we had a very short timeline, we could have one developed, an N-1 process, probably within 3 to 4 experiments. And then we might want to do some process confirmation. So I would say within a few months, we could have a good process developed for a new antibody.
Yeah. I think that's feels like the take-home message from including PAT inside it. It just changes how fast you can get to that a process that can move forward. Okay. Next question. We're going to move over to Spencer's question. How simple is it to transfer from a VVD to a CSPR process using a biomass probe?
I might see, Charles, do you want to answer this? Because you were actually part of the team at the beginning when we started implementing these probes.
Yeah, no, so I think from transferring from a volume vessel per day process to a cell-specific perfusion process using a biomass probe, it's pretty simple from just having your initial data set when using a VVD strategy, just to kind of get an understanding of what might be that CSPR min for your cell line. So you can do a couple experiments that way, just altering, a stepwise increase in volume vessels per day. But once you're able to, actually get a biomass probe, do some characterization for your specific cell line, and then using just a few data points when it comes to different CSPR levels, it's pretty quick and simple to transfer what you have from your initial data set knowledge from a VVD strategy and then applying that directly to a CSPR. So doing a VVD strategy is definitely transferable, but it's not as you've seen with Emily's presentation, it just becomes more efficient when you implement that strategy using a biomass probe.
Yeah. So, our audience it's great. The audience is very on topic around the probe and the PAT part of this, which is fantastic. So I'm going to bundle a bunch of questions around the probe itself, right? And the next question is the slope, the analytical slope that comes from the probe, is that antibody type dependent or cell line type dependent, or both? Doesn't I guess it could be both?
Yeah. So in this study, we had everything was from the same parental cell line. And they were expressing different antibodies. However, even if we, for example, had like a reclone of an antibody, so it was still expressing the same antibody, but just like a slightly different, genetic background, we did see a differentiation in slope. So I think it's basically just specifically product dependent, maybe even more specific than product. So it's something that definitely needs to be established when you're working with any new molecule, even if it's the same clone type.
Yeah. So if you see variations between clones after recloning or subcloning, then you're definitely going to see it if you move an entire cell line. Yeah.
Yes.
Most likely. Interesting. Okay. All right. So if we stay on that theme, I'm going to mark it. So if we go on that theme, so we have one from Joanna. Let me get that one up. Have you evaluated ATF for production perfusion? If so, how can a biomass probe be implemented in that process? If I read that question right, it sounds like if you scale up to production, like if we move up in volume size, can you still implement the biomass probe? And maybe are you using the biomass probe just for development, or are you using it for production too?
Yeah, so this probe, for N-1, we are using the probe, and in N-1, we see exponential growth. And the InSight probes that we use, or more in general capacitance probes, are really good at correlating, have a linear correlation in terms of the online permittivity to the offline VCD during exponential growth, because it's really measuring a cell volume. And so once you start applying it to the production phase, then things get a little bit less linear. So the the cells might be changing in diameter, but not really changing in cell number. And that can cause a little bit of noise. And so that is something that we are pursuing, but it's a little bit more complex in terms of how to model the performance of a production phase with the capacitance probe. Just it's a little bit less straightforward, less linear.
Okay.
Yeah,
So
to build off that,
I don't. Go ahead, Charles.
I was going to say, just to build off that too, using a biomass probe definitely has a place when it comes to production perfusion, such as bleed operation. But as Emily kind of alluded to, is the actual modeling may take a little bit more, data massaging when it comes to dealing with cells that may increase in diameter, looking at it from a cell volume perspective and trying to maintain that level instead of a cell number. But there's just a little bit more potential development and data analysis that may be required for that. But it does become a useful tool if you were to use it for more, I guess, efficient control of a production perfusion process.
Yes, and it is something that we are pursuing too.
Gotcha. So a question from like somebody who knows nothing about capacitance or capacitance probes, right? So is the signal only sensitive to cells? Like, so when you go into the production bioreactor, you're going to have a lot more product. You're going to have a lot more things, right? Like it's going to run for a lot longer. Do those also produce capacitance? Does that also make it harder to track VCD, or is it really specific to cells and size of cell?
My impression that it's very good at just monitoring the capacitance, or of live cells.
Gotcha.
And there's different frequencies too. There's different frequencies on the probes themselves that they can kind of cancel out some noise that you may have from other contributing factors. So just get into more of the modeling aspect. Once you get a stronger data set with these probes, you can use kind of different frequencies to better establish a model for actual live cells for your process.
Definitely.
All right, Charles, you mentioned bleeding. So I'm going to go to Zal's question on bleeding, right? So Zal's question is, do you control VCD via bleeding? And will it be based on the online capacitance reading?
Yep. So for the case study, at least that Emily presented on, for an N-1, there wouldn't necessarily be a bleeding step, since for that process, it's not a steady state. You are just growing exponential until you hit a certain cell density and then transferring into an end stage process. But for production type perfusion process, that is something you can apply capacitance probe for, and so that would just be maintaining a specific capacitance reading over the course of a long-term perfusion process. So you would have that bleeding pump directly connected, or at least connected in series with the capacitance sensor, to control actively the removal of cells to maintain a cell density that's consistent over a long period of time. So that'd be more of an application for the bleeding step, would be more end stage versus the N-1 application that Emily presented.
All right. Continuing with the capacitance probe, we're not going to pretend here like that everything works all the time. Like we all spent time in the lab. We know that's just not reality. So Christopher is just going to ask the common question that everybody wants to know, like what goes wrong. So let's just, I think it's a fair question. So what are the common failure modes? Emily or Charles, like what does go wrong, and maybe what do we do about it? Like how do you get around it when things go wrong?
Yeah, so I think some of the common things that happen is sometimes, we'll have sort of a failure of the probe to communicate with the sort of software. And so one of the things that we did that's actually in the paper that I didn't present today, if you want to look it up, is that I think a very important step to do is to sort of once you've established your online control and you do it a couple times, you'll be able to see it's very consistent, the amount of media used. So you can basically translate your optimized perfusion rate from what you have controlled online to an offline sort of VVD controlled rate. So you might be controlling one VVD over days 1 to 2, 2 VVDs 2 to 3 or whatever, but it's basically completely based on your online controlled method that was you didn't have to spend a lot of time optimizing. Now you can have that offline, and that is sort of your backup plan. So if things go wrong, you already have a defined amount of media you need to feed into the reactor over 24 hours. And that works really well. We showed that we basically see the same exact results in terms of VCD and viability with that offline control from the optimized process.
All right, so we do have a couple more questions in the queue, but we're going to make this our last one just because we're running up against our allotted time. So the last question is from John. And scaling's an important component of everything that we do. And his question is, are there plans to scale to larger working volumes? But I'm curious, I think there's more behind that question. It's how big, I think, Charles, how big does N-1 even need to go? If we just stay on the N-, I don't know if John is specifically talking about the N-1 or he's talking about N. But I think the common question is, since we're talking N-1 here, how big does N-1 even need to go?
Yeah, I think a big thing in terms of scaling is what kind of facility or demand you may require for a particular molecule. So if it's something where a company's preferring more smaller, agile facilities across multiple sites, then that may not be as large of a production scale as maybe a blockbuster drug that gets manufactured at one site and needs to be at an end stage that could be 20,000 liters. So if that's the case, then you may require a larger N-1 stage to seed that bioreactor. But a lot of it's just kind of looking back on what that end stage scale is and what VCD requirements are to seed that end stage at a particular cell density, and what does that N-1 working volume need to be. And with that being said, we've scaled N-1 processes up to 3,000 liters before, so it's something that could be needed for a particular process, but it really depends on the molecule, the demand, and what kind of manufacturing strategy a specific company may have.
Yeah, and I would just say in this study, we scaled up from 5 liters to 500 liters, and everything, worked as it should have. So I imagine scaling up even more would also work.
All right, so if we didn't answer, there's even more questions in the background. So thank you, everyone, for those questions. If we didn't answer your question live today, or actually even if we did, you will receive a written follow-up from Charles and Emily and the team. So with that, honestly, I just want to take a moment and thank Emily and thank Charles for taking the time and preparing and joining us today for both the presentation and the discussion.
Thank you, Wes. And this is a great study, or just an indication of how PAT technology can be incorporated with different types of perfusion or intensification strategies. So it's very exciting to see some of this work be presented. Very happy to have Emily here today as well to present some of that work.
Yeah, and thank you, Wes and Charles, for having me. Charles did a lot of work on this project too, actually. So I'd like to thank him for his work on here. And also, yeah, I think it's just, it was a lot of work from a lot of people in BMS. So I just want to acknowledge the whole upstream group with all of their efforts in this work to help us implement this PAT technology into a really great platform, robust platform.
Yeah, it's nice. It's making intensification even easier. We can go faster between the platform. All right, so with that, we're going to open up our poll question. If you didn't have an opportunity to answer our poll question in the beginning, it's going to be live up now. Otherwise, again, if you want to go back and learn about other case studies, there's multiple other webinars at repligen.com/resources where you can learn about the technology, vaccines, viral vector intensification, all sorts of things there. So be sure to check that out. And otherwise, I will see you next time for our next episode. Take care.